inside, it's a wild ride. Come on! Ride on the magic school bus.
Good morning, class. I'm Mrs. Frizzle. Today we're going to be talking about targeted nanodelivery.
What's targeted nanodelivery?
Arnold, you don't know what that is?
Guys, let Miss Frizzle talk. Let's get the facts.
Targeted nanodelivery is a method that uniquely is capable of localizing delivery of therapeutics and diagnostics to diseased tissue. This method reduces the quantity of drugs required, is safer for non-targeted tissue, and is a great method to deliver toxic drugs. And we have a field trip!
I knew I should stay home today.
You're such a wimp.
Hey guys, it's all okay. Let's hop on the bus. We're gonna go to the doctor's office today, where Mr. McLean, our chain-smoking janitor, is gonna get a checkup because this boy was coughing up blood today.
Woo! I think I'm gonna be sick.
You're a wimp too.
Let's go!
Hey kids, I was just injected with some particles called nanoshells. Does anyone know what nanoshells are?
I definitely wouldn't know. My dad's only a major researcher at a top research university.
Did he write about your tiny brain for his thesis too?
Shut up, Ralphie.
You're respectful, kids.
I've got a book on it. Nanoshells are spherical cores of a particular compound surrounded by a thin layered shell or outer coating of another material. Nanoshells are highly functional materials showing modified and improved properties compared to single-component counterparts or nanoparticles of the same size. Their properties are modified by changing either the constituting materials or core-to-shell ratio. Nanoshells have improved optical properties and reduced susceptibility to chemical thermal denaturation compared to conventional organic dyes. For applications involving imaging, gold is often the material of choice when designing these nanoshells.
Thank you so much, Keisha. That was very insightful. Be respectful, kids.
Hey, actually, I know what nanoshells are too now. I think I heard about it once because they were doing a study where they injected some into the body so they can tell where the tumor is.
Oh no, that sounds scary.
Oh, so we got to inject him with a drug like my cousin does in his spare time. Relax, guys, he has diabetes.
Well, let's go into his body just to see the interaction of these drugs with the cancer cells.
Whoa, Miss Frizzle! What's that? Look outside the window! What is that?
Oh, look out the window, class! It's a nanoparticle functionalized to the surface with antibodies. It's designed to target a site. It's called active tumor targeting.
What? How does that work?
It can specifically target tumors because the surface has been functionalized with a molecule called an antibody. Antibodies are proteins that can be designed to specifically recognize proteins in the body. Sometimes cancer cells may have a particular protein on their surface, and using antibodies and binding them to those proteins, we can guide these molecules to the cancer.
Wow, so it's almost like a magic bullet.
Get it? Because he got a shot.
Get away from me.
Hey guys, what's that small crystal past the window?
That, my dear Arnold, is an exciting type of nanoparticle called a quantum dot.
Oh, what a dot?
I've definitely heard about quantum mechanics at the age of 9, but
Yeah, me too.
I'm not sure what a quantum dot is.
It's okay, Ralphie. Quantum dots are semiconductor nanoparticle crystals. They trap electrons in a potential well. Can you guess how they can be applied in a biomedical context?
No, we're only 9 years old.
Come on.
Alright, folks. Many biologists are working on quantum dots to tag individual molecules so that scientists can track cellular processes. Therefore, quantum dots are helpful for medical diagnostics and targeting tumors for the delivery of anti-tumor nanomolecules. Let's watch it bind to a cancer cell right now. Put your seatbelts on, kids! Whoa! Quantum dots can do much more than that. Quantum dots also can help to facilitate high-resolution imaging within the cell, which means they can replace traditional fluorescent dyes that were used for imaging. Anyone know how these quantum dots can be fluorescent?
Maybe like other fluorescent dyes, do quantum dots excite their electron to higher energy levels and drop them back by emitting photons?
Almost right on the dot, Keisha! Do you know how they can be excited?
Not really. My dad might have told me a lot, but I'm still in elementary school.
Well, pay attention, young lady. The quantum dots are excited with light, and when that happens, the electrons with the quantum dot are excited from their valence band to the conduction band. Upon relaxation of the electron from the conduction band back to the valence band, a photon is emitted. The emission of quantum dots can even be controlled by their size.
Oh, I see. And it's excited because the light carries energy. So it's almost like it's pushing the electron up a flight of stairs, and once the excited electron falls down, the light is emitted.
Exactly. But Mrs. Frizzle, how these quantum dots can replace traditional dyes? Are they better than original ones?
Good question, Arnold. Because of this high extinction coefficient, it has been estimated that quantum dots are 20 times brighter and 100 times more stable than traditional fluorescent dyes. Not only that, because of their versatility, quantum dots have been used in vitro imaging for applications in embryogenesis, cancer metastasis, stem cell therapeutics, and lymphocyte immunology.
Mrs. Frizzle, I don't get it. What are these nanoparticles all made out of? They can all be made out of the same stuff, right? Like, the nanoshells are gold and all, but none of these particles look really shiny to me.
All that glitters isn't gold, and vice versa. The reason they don't look gold, Arnold, is because of the nanoscale. The wavelength of light that's reflected is different from the macroscopic world. That's why sometimes if you have a solution with gold nanoparticles, it looks red.
Very good, Ralphie. You're right that the gold is not really the color we expect when we see it on a large scale, but you don't answer the original question of the material of these particles. The truth is, Arnold, that a wide variety of materials are used in the creation of these nanoparticles, as well as a wide variety of techniques. This gives researchers a large toolbox to work with when designing these tiny machines to seek out and destroy cancer.
Hey, Miss Frizzle, isn't silica a really popular material to make these particles out of?
Actually, I can answer that question because my dad is a researcher for Bang's laboratory. So let me drop some knowledge on you. So silica is a really, really popular material, as you said, for making nanoparticles because silica is really good for biomedical applications. It has high biocompatibility, low toxicity, ease of availability, thermal stability, but most importantly, there's a wide range of functionalization techniques that you can use on silica microparticles.
Wow,
and nanoparticles.
That's really interesting. Could you explain that last part a little bit?
Yeah, actually. My dad's a chemist and he taught me quite a bit about surface modification techniques. So let me drop some knowledge real quick. What Wanda meant is that there are a lot of different techniques that someone can use to change the surface property of a particle. For example, scientists can use a chemical called aptase, a siloxane, which functionalizes the hydroxylated surface of a silica nanoparticle into amine groups, which can then be bound to protein. Other techniques can be used, so not only could you bind proteins to a particle surface, but also oligonucleotide chains to specifically bind free DNA or RNA single strains in serum that may serve as a biomarker for disease. There are also particles that are mesoporous, that is, it contains pores between 2 to 50 nanometers, and these can be loaded with drugs that can then diffuse out of the particle core when the particles are endocytosed. Look, there's a cell endocytosing a particle right now.
Wait, Ralphie, when did you get so smart? Also, what is endocytosed?
You're not that smart either, Arnold. Weren't you paying attention during 3rd grade cell biology? Endocytosis is when particles are uptaken by a cell. This can be influenced by a particle's charge, due to the inherent negative charge of the cell membrane. Therefore, positively charged particles are better at binding and being taken in by a cell. Most importantly, however, is clathrin-mediated endocytosis.
I think it's coming back to me now. Clathrin is a cell membrane protein that is involved in recognizing molecules outside the cell and bringing them inside the cell if deemed appropriate, right?
Right. There are other pathways too, like caveoli-mediated endocytosis, that involves caveolin-1 expressing structures, and second cholesterol-dependent pathway, macropinocytosis, which is involved in fluid phase endocytosis, and where the endocytosed molecule or nanoparticles are located in micropinosomes. What's important to take away, though, is to design particles with these strategies in mind, so your particle and the drug payload can get inside the cell.
Wow, this is a lot to take in. Just like Mr. McLean's cigarette smoke, am I right? Also, speaking of which, we're at the lungs, where the tumor is. And look, it's those targeting molecules with the antibodies we saw earlier. Oh wait, this is Frizzle. What are those branch-like things?
Oh hey, Mrs. Frizzle. I think I see what Arnold was talking about. It looks like that spiny hedgehog over there.
That, my dear and curious student, is a dendrimer. Like you mentioned earlier, Arnold, it does indeed have a branching structure. The Greek word from which it is derived actually means tree.
Wow, so interesting.
So, what are they doing exactly?
Do our resident nerds Wanda or Keisha, as well as surprise nerd Ralphie know?
Nope, never seen one before in my life.
Hey, Arnold!
Ouch!
Ralphie, stop putting Arnold in a headlock.
Now, dears, play nice.
Okay, Arnold, surprise nerd talking now. Dendrimers are highly repetitive branched molecules. Polyamidamine dendrimers are the most attractive variety for cancer treatment nowadays, since they have an exponential branching growth, easily functionizable groups, high monodispersity, and an extremely large number of surface sites relative to volume. Not to mention, they're cheap to synthesize and are extremely biocompatible.
Sounds like a terminator of nanoparticle drugs.
And that's not all. Polyamidamine dendrimers derivatized with folic acid are preferentially absorbed by cancer cells as they overexpress the folate receptor on their surfaces. This allows the ligation of additional treatment methods to the dendrimer, like methotrexate, boron isotopes, and cisplatin that can release its cargo following its intake into a cancer cell. These treatments then attack the cancer cells at their source, preventing collateral damage to other cells in the body. Look at them go!
Whoa! What?
Now the tumor is dead. Can we go on now?
Come on.
Yes, Arnold, we can finally go.
Boo.
And on the way, you can all start on your next assignment. A get well card for Mr. McLean.
Okay. I'll buy cookies.
What a trip, guys, am I right?
That was so interesting. We learned a lot about drug nanodelivery today. Thank you, Mrs. Frizzle.
Thank you.
You're right. One thing I was wondering though, how did smoking cause Mr. McLean to a cancer?
Wait, Miss Frizzle, let's get the facts.
Okay, take chances, make mistakes, get messy. Let's find out, kids.
Yes!
Let's get on the bus, boys. Oh,
Yes!
and girls.
Bus time!
I'm gonna be sick.
I knew I should stay home today.
Do your stuff, bus.
Surfing on a sound wave, swinging through the stars. Take a left at human testing. Take your second right past Mars on the magic scuba. Navigator nostril. Climb on the magic stool.